Hi, in this video we're going to talk about how B cells are activated with the help of T cells. So this process is called the T cell dependent activation of B cells. Let's start off by first talking a little bit about the B cell. Here we have a B cell with its B cell receptors, which are going to be antibodies, specifically the antibodies IgM or IgD. In this image, we have the receptor as IgD because it simplifies the image of the B cell. Some other things to know about B cells is that B cells are part of your adaptive immunity, along with T cells and antibodies. Stem cells from red bone marrow differentiate to become B cells. B cells mature in the bone marrow. However, you could also find mature B cells in the lymph nodes and throughout the blood. In order for a B cell to be activated with the help of a T cell, the antigen present must be a T cell dependent antigen, which are going to be proteins. And these are going to be proteins of the virus, the bacteria, or whatever agent has invaded the body. Here we have our protein antigen, and the different shapes represent the different epitopes that the protein antigen will have. An epitope is a specific area of the antigen that can be bound to. And protein antigens have a lot of different epitopes. And in this image, each different shape represents a different epitope. The process starts when a B cell, using its B cell receptor, binds to a specific epitope. The whole antigen enters the B cell. The antigen is then broken up and then combined with major histocompatibility complex 2, simply put, MHC2. The antigen MHC2 complex migrates to the surface of the B cell. This is why B cells are considered to be antigen presenting cells, because they're able to show or present antigens on their surface for other immune cells. Thanks to MHC2, the B cell can present the antigen. So now in the lymphoid tissues, the B cell looks for a helper T cell that can bind to one of its antigen fragments that it's presenting. This is one helper T cell that can activate this B cell because it can bind to this specific fragment. Once a helper T cell binds to the antigen fragment that it is specific for, the T cell then releases cytokines which activate the B cell. The activated B cell makes a large number of clones of itself, and some of these clones differentiate to become plasma B cells or memory B cells. The plasma B cells secrete a lot of antibodies which help fight off the pathogen. The memory B cells are there so that if you encounter the pathogen in the future, your body can then mount a quicker immune response against that pathogen. The response will also be more effective against the pathogen. When plasma B cells secrete antibodies, they first make IgM. Depending on the location of the infection, IgM might not be the most ideal antibody to have. So, for example, an infection that is located in the mucous membranes, IgA would be ideal.
Fortunately, we have class switching. Class switching is a process that occurs after a B cell is activated to enable the plasma cells created by the B cell to be able to make different classes of antibody. So here's how it works. So first, we have a helper T cell that binds to the antigen fragment presented by the B cell that it is specific for. Once bound, the helper T cell releases cytokines to the B cell. So now the plasma B cells produced by this B cell can secrete the necessary antibody, which would be IgG, IgA, or IgE. You may notice that IgD is not listed, and the reason is because mature B cells can actually secrete IgM and IgD, and so there's no need to class switch to produce IgD. That's pretty much it for this video. I hope that you saw just how important your helper T cells are because without your helper T cells, your immune system is reliant upon the T cell independent activation of B cells to really fight off infections, and that's not going to be as effective as the T cell dependent activation of B cells to fight off infection, because with the T cell dependent activation of B cells, you can have class switching occur, right? So that you can produce the needed antibodies for whatever infection is going on. And you also have plasma and memory B cells that are produced. Recall that memory B cells allow you to be able to fight off the pathogen by mounting a larger and a quicker, more effective immune response against the pathogen the second time your immune system encounters it. Thus, you have a stronger immune response. However, if you don't have any helper T cells, your body is limited to T cell independent B cell activation, which results in mostly IgM being produced, which is not going to be ideal for every type of infection that occurs. Also, you end up with a weaker immune response because no class switching occurs, you have no memory B cells that are produced, and IgM is mainly the antibody that's produced. That's it for this video. I hope this video helps. Please like, subscribe, comment, and share.